Okay, so today's talk is going to be on transposons, transposable elements, also called jumping genes. So these have been widely considered to be selfish DNA or junk DNA. Anytime you hear someone talk about all these copies of things in the genome, they're usually talking about either repetitive DNA, which we've discussed before, or they're talking about either repetitive DNA or they're talking about transposable elements or insertion sequences. So, so that's what we're going to cover here. And far from being junk DNA or selfish genes, these genes actually they're highly regulated. The transposition mechanism is highly regulated, and in fact, we actually have examples of these happening um, in specific response to selection. In fact, this has actually been known since the 90s. It's just not often talked about for reasons discussed in this paper. So this is a 2015 paper. This is by Sayer and Zhang, and those two guys have done a lot of research on this, and so you can. I suggest you kind of look those guys up. They've got a lot of papers discussing the specifics of these sorts of mechanisms, and well worth your time. So here is a specific instance of a jumping gene that is controlled by selection pressures. Okay, so you will get the insertion sequence if the selection pressure occurs in the way that it matches, and you will not get it if it doesn't. So, all right. So this is the see if I can pronounce this right. Phosphoenolpyruvate sugar phosphotransferase system. All right. All right. So basically, there is an insertion sequence called IS5 that hops into a specific site upstream um, of this gene in E. coli. All right, so it activates the expression, allowing growth of wild-type cells with glycerol as a carbon source in the presence of non-metabolizable glucose analogs, which normally block glycerol utilization. The sugar-transporting PTS controls this process by regulating levels of cytoplasmic glycerol 3-phosphate and cyclic AMP as established in previous publications. Direct involvement of the glycerol repressor and the cyclic AMP receptor protein in the regulation of transposon-mediated directed mutation has been demonstrated. Okay, so let's back up a little bit. Let's talk about insertion sequences. So let's see here. Um, so transposons are jumping genes. So they basically say that they break into two kinds. They're either going to be insertion sequence that can be kind of replicated all over the genome. Let's see here. They usually recognize and insert into specific target sites in host DNA. Let's see here. It can occur either by a replicative mechanism, in which case a new identical element is synthesized, or by a non-replicative mechanism, in which case the element is cleaved out of its original site and transferred elsewhere. It can inactivate a gene, activate expression of an operon, and so on. So here we're talking about a specific one called IS5. And so the protein is INS5A, and basically there's a binding site and a DNA bend. So here's how it works. So basically there's a target site, and the transposase gene is flanked by two inverted repeats. That's what the IR stands for, is an inverted repeat. And so basically the transposase recognizes the target site, it duplicates the target site, see how that's on both sides, and then inserts the insertion sequence, which has these flaking sequences. So just from an off-the-cuff engineering perspective, so it looks like this is done so it can actually uncleave them as well, so that in the future, it can recognize, first of all, it can recognize this matchup right here will tell it that it's already been inserted at that location. Possibly, so if there needs to be prevention of inserting it again, we know that we already have that one. Second of all, is that if you have this forward and backward, it's invertible repeat segment, and these are pretty short. Then you can use that if there's another protein we could use to remove this insertion sequence as needed. So anyway, very interesting system. So the insertion sequence 5, just to give you a little bit of background information on it. So let's see here. It is about 1,200 base pairs long, and the inverted terminal repeats are 16 base pairs. So those inverted repeats on the ends, there are 16 base pairs. The target site for IS5 is 5' CTAG. So anyway, it's a really interesting gene. So okay, so this also gives a little bit of background on jumping genes and actually on, let's see here. That's the phototransferase system. It's a little bit about Darwin and Lamarck. I don't like to mention Darwin very often just because I think we've moved past him, and hopefully, there's a lot of references to him and try to working things out so it matches him. And I'm just like, why are we even having that discussion? It doesn't seem to be relevant. So anyway, so here's the proposal of directed mutation. This is an interesting history on directed mutation controversy, and it's interesting just that how much, even today, the idea of directed mutation is still widely resisted despite all the evidence for it. So let's see here. So they give several examples of directionalized mutations. So here again, they're talking about the dogma of our time. However, it goes against the dogma of our time. And the dogma of our time states that mutations occur randomly and that the advantageous mutations are selected only after they arise. And it says a concept that is in detail now known to be inaccurate. The notion of directed mutations has been highly controversial and not generally accepted in the scientific community even today. All right. So anyway, despite the fact that they're giving direct examples, this is still not widely known and is still not widely accepted. In fact, there's actually kind of a frustrating thing. Let's see if I can find where it was. So here's the evidence for it being directed. So when these mutant cells are placed on minimal glycerol plates, insertion sequence 5 insertion mutations arrive after a lag period during which cells are starving. So the insertion sequence don't start arising until after the cells are starving. Starvation in the presence of glycerol. All right. So if you starve them and put them in the presence of glycerol, it activates the hopping of IS5 to the activating site. All right. So you have starvation in the presence of the thing that you're wanting to metabolize, and it will cause the insertion of this sequence into the right site, which relieves the stress. And so numerous control experiments have led to the conclusion that this activation event is specific for this one insertional site in front of the promoter. So this is the one. This is what I was thinking about earlier. So this is frustrating to me. It says this specificity could have been selected for by currently recognized mechanisms of Darwinian evolution. That sentence actually, there is literally they don't have any. This is a sentence that doesn't have any experimental data. There's no experimental support. There's no mathematical support. It's just dropped in there as an as an homage to what the wider biology community thinks. There's no reason for it. It doesn't function anywhere in the paper. It doesn't tell us anything more. It's just there to make people feel better. And I don't know if it's the authors feel this way. That could be. It could be that this is what the authors think, or it could be that it was reviewers like, well, what about Darwinian evolution? Um, or it could be the editor. I don't know where in the chain this got added, but it seems just out of place because literally there's no evidence for it. There's no reason it needs to be there. There's no requirement for it. It just gets dropped in. So anyway, they go through the mechanism of the binding sites and how they went and figured this out. And so anyway, it's really fascinating. So insertion sequence 5. But the interesting thing is that this, even though the specifics of the mechanism weren't known, the general idea that there's insertion sequences that get added in stress events has been known since the 90s. So this is an interesting paper from 1999, Transposable Elements as Activators of Cryptic Genes in E. coli. All right. So the abstract says the concept of transposable elements as purely selfish elements is being challenged, as we have begun to appreciate the extents to which transposable elements contribute to allelic diversity, genome building, etc. Despite these long-term evolutionary contributions, there are few examples of transposable elements that make direct positive contribution to adaptive fitness. So basically, we haven't found all the ways, we haven't always mapped how they're specific to helping, we just know that they're there. So, but there are some examples of transposable elements that make positive contribution to adaptive fitness. So here, basically, catabolic operons can be activated by small transcriptional element or transposable elements that called insertion sequences. Not only do insertion sequences make a direct contribution to fitness, by activating cryptic operons, they do so in a regulated manner, transposing at a higher rate in starving cells than in growing cells. In at least one case, insertion sequence elements activate an operon during starvation only if the substrate for that operon is present in the environment. All right. So anyway, in this paper, I used to have it. I don't know where it went. But it's a great paper, and it's basically I believe it is beta-glucoside sugars, and it is the promoter is IS1 and maybe IS5 as well. So IS1, just to give, let's see here, IS1 contains 768 bases, and about 30 bases at the ends of IS1 were found to be repeated in an inverted order. All right, so you've got the 768 bases with the inverted repeats on the end. So anyway, it's so kind of the same basic mechanism. So as far as I'm aware, no one's actually gone through the trouble of figuring out exactly how this one works, which is weird because it's been there. We've known about it for literally over 20 years now. So it's just amazing to me how little attention this gets. So anyway, that's transposable elements. Not only do they happen in non-selective places, you have increased transposable elements when an organism is under stress, and you have some transposable elements that go to very specific places under very specific stresses. So anyway, so I hope that's interesting for you all.